cancer in general, we think about it being diagnosed at an older age. Also, things like a person's reproductive history, how much dense breast tissue they have, and lifestyle factors, smoking, drinking, alcohol exposure, exposure to cancer, like carcinogens, so things that increase the risk of developing cancer. All of those things overall increase your risk. But what we know, and from our perspective in genetics, is that a person's family history provides the greatest risk that is associated with an increased risk of developing breast cancer. So the more people there are in a family that are affected, the higher the relative risk for that person. And based on these risk factors, there is screening that has been recommended for the general population. So this includes monthly self-breast examinations, clinical breast examinations, and mammograms after the age of 40. So when we take a closer look at breast cancer, I started off by saying that breast cancer is really common and it affects 1 in 10 females. But when we look at those breast cancers, we can split them up. So we know that in about 5 to 10% of all the breast cancers that are diagnosed, there are some that are believed to be hereditary. So what this means is that they are caused by the transmission of faults, or we call them mutations, in a person's genetic information. And these mutations, so having a mutation increases your risk for developing breast cancer, as well as other associated cancers, depending on what type of genetic fault you have. The rest of the cancers, so the other 90 to 95%, are what we call a combination of some being familial and the rest being sporadic. So what we mean by sporadic is that they are cancers that occur by chance. There's not one specific thing that we can pinpoint to be the cause that causes the cancer or increases the person's risk for developing cancer. So we know that it probably is a combination of lifestyle factors and your family history all working together. But when we try to look for a genetic cause, we can't find anything. So genes, I mentioned on the previous slide, which are the instructions that tell the body how to perform specific functions, are what we see being impacted in hereditary forms of cancer. So there are specific genes that have functions in the body, and when people have genetic faults in those genes, it can cause a person to be at a higher risk for developing cancer. So I will explain this now in more detail. So there are several genes that have been found to be associated with an increased risk for developing breast cancer. And we know that cancer arises from the uncontrolled growth of cells in the body. So our bodies are made up of millions and millions of cells, and each of those cells has to follow a set of rules that are in place. A cell knows that it needs to start growing, grow to a certain point, and stop growing. And that is what we call cell cycle control. There's control in how our cells are growing. But sometimes, if we have a fault in a gene that is helping to regulate how cells grow in the body and how cells function, it means that the body is under less control. There's less things that are trying to protect cells from growing abnormally. And so normally what we see in cancers is that there are faults in genes that are helping to control how cells grow in the body. And so the resulting cancer happens because those cells have now grown out of control and are disobeying the rules in the cells. So mutations or genetic faults in these genes increase an individual's risk of developing breast cancer and other associated cancers. But the associated cancers that are at an increased risk depend on what type of gene is involved. So if we look now from a bird's eye view, we know that there are 90 to 95% of breast cancer cases where if we have to investigate them, we can't find a known genetic mutation. But there are about 5 to 10% of them where we know that there is a genetic mutation that is involved in this cancer somehow. Of these, they can be caused by mutations or faults in high-risk genes. And I'm sure these BRCA1 and BRCA2 genes are genes that are very common. You come across very often when you read about breast cancer, and I'm sure a lot of you would know about them. But there also are other genes which are called moderate risk genes. And these genes do increase a person's risk for developing breast cancer and other associated cancers, but the risk is not as high as it would be for the higher risk genes. Okay, so an important comment to make, and I think here is probably a good time, is that there is a difference between what we call germline and somatic mutations. So germline mutations are genetic faults that an individual is born with. So these are mutations that have been inherited and passed down in a family over many generations. When we see people for breast cancer counseling consultations, a lot of times they tell us that they've had genetic testing done. And very often what they're talking about is somatic mutation testing. So the difference is that somatic mutations are genetic faults that occur in the genetic information of the cells that make up the tumor. So that's what we call tumor genetics. So they take a biopsy of a cancer and they send it to the lab, and the lab does genetic testing on that tumor. And because we know that cancer occurs because cells are growing out of control, we expect to see genetic faults or changes in the cells that make up the tumor. But what that means is that having a somatic mutation or a mutation in the tumor does not necessarily correlate back in saying that the cancer is hereditary. So having a mutation in the tumor doesn't mean that you necessarily were born with that mutation. That mutation might have been something that happened in your body during your lifetime. But what the tumor testing is important for is helping to guide treatment and management in some cases. Okay, if anyone is very lost or confused or needs me to repeat something, please shout out. I don't want anyone waiting right until the end. Okay, looking at the genetics of breast cancer. So we are going to focus more on the genetic aspect now. Genetic information, I said, is inherited. So we get half of our genetic information in the egg from our mother and half of our genetic information in the sperm from our father. So what this means is that every person should have 2 copies of each of their genes, one that is inherited from each parent. So for every gene that we spoke about, I mentioned the BRCA1 and the BRCA2 genes, but there was a list of genes that we know are all associated with an increased risk for developing breast cancer. And what that means is that for every single one of those genes, we have 2 copies. We have one that is what we call maternally inherited, so inherited from our mother, and one that is paternally inherited, that's inherited from our father. So I know a lot of times you hear people saying, I want to find out if I have the BRCA gene or I have the cancer gene. We all have BRCA genes. Every single one of us should have 2 copies, one that we inherited from our mother and one that we inherited from our father. What we're looking for when we're doing genetic testing is to see if either one of those copies has a fault in them. So mutations that are associated with an increased risk of developing breast cancer are inherited in what we call a dominant manner. So what this means is that a person only needs one copy of a faulty gene in order to be at an increased risk of developing cancer. So if we had to look at one of these breast cancer genes, if you have a mutation in one of those genes, then you are at a higher risk for developing breast cancer. You don't need to have both of them mutated, you only need to have the one. Again, having a mutation does not mean that you will develop cancer. It just means that you are at a higher risk. So the risk is increased, but not 100%. Okay. So looking at dominant inheritance a little bit more closely. If you have two parents, for this example, that both are wanting to find out what their risks are of having a child with one of these high-risk mutations, and let's say we've tested one parent and the one parent has come back saying that they have a mutation or a genetic fault in one of their genes that increases their risk for developing cancer. When you have a child, you pass on half of your genetic information. So this parent will either pass on this instruction with the mutation, or they will pass on this instruction with a normal functioning copy of the gene. The other parent has got two instructions, one that they inherited from each of their parents that have normal functioning copies of the gene. So regardless of which one this parent passes on, they will always pass on a copy of the gene that is functioning properly. So if we look at it, there are 4 possible combinations that can come out of how these parents pass on their genetic information. This is the first, where the first parent passes on the instruction with the mutation and the other passes on one of theirs. Or both parents could pass on instructions that don't have any faults in them. Again, now this parent passes on the one with the faulty gene, and this parent passes on the other instruction. And then in the last example, both again pass on 2 other instructions. So you can see from this example that every time a person with a genetic mutation that is dominant, like the ones we're talking about today, every time they have a child, they have a 50% chance or 2 out of 4 chance of passing this mutation onto their children. So we normally say that a person with a genetic fault, so let's say, for example, they have a fault in the BRCA1 gene, they have a 50% risk of passing that same mutation onto their children. Okay, so now that we've spoken about BRCA, in this example, we can talk a bit about hereditary breast and ovarian cancer syndrome. So this is a syndrome. Syndrome just means that it's something that's genetic, and we use the word syndrome when we see a group of very similar features that we can clump together. So hereditary breast and ovarian cancer syndrome, or HBOC for short, is caused by mutations in the BRCA1 and BRCA2 genes. So regardless of whether the person has developed a cancer in their lifetime or not, if you have a mutation in your BRCA gene, you are said to have hereditary breast and ovarian cancer syndrome. And these mutations, like the example that I just used, are inherited in an autosomal dominant or a dominant inheritance pattern. If you have one of these mutations, you are at an increased risk of developing breast and ovarian cancer if you're a female, and an increased risk for developing male breast and prostate cancer. The risk again is increased, but not 100%. And there are other associated cancers, in this case, particularly cancers like pancreatic cancer and melanoma. So I'm going to show a table and just this comment just to say that individuals with the same mutation may develop different cancers. So you could see someone with a family history where there are many people in different generations that all have the same BRCA mutation. You could see a mother and a daughter, and they may have completely different cancers. Okay, so this table is just to illustrate how the risk increases compared to the general population if you have a mutation in BRCA1 or BRCA2. So if we look at the first row, the risk for female breast cancer in the general population, we said about 1 in 10, so 10 to 12%. If you have a mutation in BRCA1, that risk goes up to 87%, and in BRCA2, up to 84%. There is also an increased risk for having a second primary breast cancer for people who have already had one cancer. And there's an increased risk for ovarian cancer. So in the general population, the risk is 1 to 2%. But with the BRCA1 mutation, your risk goes up to 63% and slightly less for BRCA2, which is 27%. However, with BRCA2, the highest risk for male breast cancer is with BRCA2 mutations, as well as the prostate cancer risk. And then this last row just summarizes some of the other cancers that are associated with these mutations. So even though there is such a high risk with mutations in these genes, it's not, and we as in genetics never want to go to someone and say, you have this mutation and good luck with your life. The point of doing this testing is that because these genes are so common and these mutations are so common, and we know so much about them, is if we can find a mutation in a person in one of these genes, and not just the BRCA genes, but I'm using them as an example now, is that there are very well-established management and screening guidelines for people to follow as recommendations based on what we know about the mutations in these genes. And so by knowing their genetic information, we can help guide them on how best to manage their risk. So the recommendations are for individuals that are at high risk of developing breast cancer. And we know that with BRCA mutations, people are at a higher risk. In fact, the BRCA genes confer the highest risk for breast cancer out of all the genes. So the recommendations for breast cancer in terms of screening are monthly self-breast examinations, also annual clinical breast examinations, and then annual or biannual mammography and or MRI, breast MRIs. I'm very aware that these options are not available to everyone across all state and private services. And then the recommendations for ovarian cancer are routine gynecological screening. But what we know about ovarian cancer screening is that it's not very effective. So breasts are on the outside of the body and they're easy to get to and easy to screen, whereas a person's ovaries are inside their bodies and a lot harder to get to and accurately screen. And so what we see a lot of the time, unfortunately, is that once an ovarian cancer has been detected, it's quite advanced. Okay, so there is also the option of prophylactic surgery. So prophylactic surgery is risk-reducing surgery that a person has prior to the development of cancer. And this is an option for many people because having something like a risk-reducing mastectomy decreases the breast cancer risk by 90%. Can never take away the risk, but being at a risk of as high as 87%, a lot of people feel that they would rather have their breasts removed because it decreases their risk significantly. The other comment is that there's a risk-reducing oophorectomy, so having both ovaries removed, and normally they do a salpingo oophorectomy, so they remove the fallopian tubes as well. And this decreases the ovarian cancer risk. Sorry, this is supposed to say percent, 80 to 90%, But also having the ovaries removed decreases the breast cancer risk by 50% if it's performed premenopausally. So these are recommendations and these are guidelines based on what we know about these mutations in these genes. But we know that it's a lot more than just that. These are very, very challenging and difficult decisions, and very often a decision that people need to work through over a period of time. So a lot of people go through the process of perhaps having a mastectomy, but then choose to do an oophorectomy later in their lives once they've had children. And it's a very personal decision for each person. So when we look at a family, we want to assess the family as a whole. A lot of times we see one person in our genetic counseling consultations, but we take their family history. And we want to look at the family as a whole to see if we can pick up any of these warning signs of hereditary breast cancer. So just looking at this image over here, this is what a family pedigree looks like. We draw males as squares, and we draw females as circles. This just allows us, it's an internationally standard way of drawing family trees. And this means that if we had to see a family tree from someone in India, we would know what they were talking about. The people that are colored in are typically the people that are affected for whatever condition it is. So when we assess a family history and we see cancers where they are an early age of onset, typically cancers that are people that have been diagnosed at an age younger than 50 years, or when we see bilateral breast cancers, when we see people that have more than one primary cancer. So let's say, for example, someone that's developed a breast cancer and then an ovarian cancer at a later age where it's not because it's metastasized, but it's because it's a brand new cancer. Also, where we see people or families where there are several family members affected. So they can either have the same type of cancer or related types of cancers. So we spoke about hereditary breast and ovarian cancer syndrome. And we know that there is a risk for male breast cancer, prostate cancer, and also female breast cancer and ovarian cancer. So if we see males with prostate cancer in a family and females with ovarian or breast cancer, even though they aren't all the same types of cancer, they're what we call related cancers. And also when a person has a lot of first-degree relatives, so siblings, children, or parents with cancers, then we also need to keep that in mind. Any family where there is male breast cancer, because it's rare, we consider doing genetic testing, or this is what we would call a warning sign for hereditary breast cancer syndrome in the family. And then any person that comes from a founder population. So we know that the Afrikaner and the Ashkenazi Jewish populations are what we call founder populations. And what that really means is there are populations that at one point in time decreased significantly in size. And so there were a small number of people in these populations and there wasn't a lot of genetic variety amongst these people. And so when the population groups grew again, there is less variety in the large group in terms of genetic information. So what we see is that when we look at the genetic information of people that belong to these founder populations, there are certain mutations that are very common in these populations compared to elsewhere. Okay, so what testing do we have? But first, the types of testing. So you can undergo diagnostic testing. And this is typically testing that is aimed at testing affected individuals. So someone that already has a cancer, and a negative result does not exclude a genetic cause. So whenever we see someone for genetic counseling and we offer them diagnostic testing because they have a breast cancer or ovarian cancer, for example, we explain to them that when we do this test, it can come back with one of three possible outcomes. Can either come back positive, meaning that we have found a genetic fault in your genetic information that explains why you developed cancer and why there is an increased risk of cancer in the family. It could also come back negative. If it comes back negative, it means that we've excluded the faults that we've looked for. So if we're only looking in 2 genes, we've excluded the faults in 2 genes, but we haven't excluded all possible genetic causes for that cancer. And it could also come back as what we call an uncertain result, where they found a genetic fault in the person's genetic information, but it could be a brand new fault, it could be something that's very rare. And so when we see it, we don't know if it's what is causing the cancer, or what is putting this person at a high risk of cancer, or if it's something that we call benign, something that's just part of normal genetic variation, because all our genetic information is a little bit different. And then there is predictive testing. So this testing is testing that is offered to unaffected individuals, but who are at risk of having a genetic fault. So this is because they have a known mutation in the family, and they have to go through what we call a predictive testing process. So let's say, for example, a lady is tested and we find a genetic fault in one of her genes. So let's say a BRCA1 mutation. And she wants her daughter to now be tested. Her daughter is unaffected, but she's at increased risk because we know her mom has the mutation. If we think back to the dominant inheritance, we know that her mom, there's a 50% chance of her mom having passed that mutation onto her. So because she's unaffected, she goes through this predictive testing process. We are only going to test the daughter for the mutation that we know is in the mom. So her testing outcomes are either going to come back positive, meaning that she has the mutation that was in the mom, or it's going to come back negative, meaning she didn't test positive for that mutation. But we're not testing for all possible faults. Now, predictive testing, just to carry on with that idea a little bit more, you can see in this family, again, the females are circles and the males are square. In this family, there is a female that has breast cancer, has had breast cancer and ovarian cancer. There's also another female that had breast cancer and a male that had prostate cancer. And so this person we tested, and this plus is here because we found a positive mutation in the family. So when we think about predictive testing, a person's genetic results can sometimes have benefit for them, but also a lot of it benefit for the other people in the family who are at risk of having inherited the same mutation. Because again, this father shares genetic information with all of his children, which means all of these siblings share genetic information, which means there's a risk that all of the children in this generation also share genetic information. And so when we look at all the people that could have inherited this mutation, there's a lot of people in the family that we could now consider offering predictive testing to. We would normally offer unaffected siblings first. So if the sister or this brother tested positive, we would then test their children. Okay, but predictive testing is not always straightforward and not always easy. And that's because there's a lot of uncertainties. Knowing that you have this mutation still doesn't tell you whether you will develop cancer or not. Remember that the risk is not 100%. And so you could know that you have a BRCA mutation, for example, but you won't know if you will get cancer or not. And if you do, what type of cancer. You also don't know what age it will develop at or how severe it will be. And it's also difficult to take into account the effects of environmental factors and other genetic factors that might modify this person's risk. So living a healthy lifestyle. All of those things are important regardless of whether you have a risk or not, but those things we can't quantify, we can't put a number to them. So we know that the person's at risk, but how do they best manage their risk going forward? Okay, so testing, we know that there's diagnostic testing and we know that there is predictive testing. We also know that people in this country are either seen through the state hospital system or they're seen in private. In state, typically we have testing for founder mutations. So these are mutations that are seen commonly in certain populations. So there are founder mutations in the Ashkenazi Jewish population. There's 3 of them that we test for in this country. And there's some in the Afrikaner population. But also we offer full sequencing of the BRCA1 and BRCA2 genes. And at the moment, that is all we offer in state. Our department has been busy with a research project trying to put together a gene panel of genes that are associated with an increased risk for developing cancer. It was in production at the end of last year and part of a research project, but hopefully towards the later half of this year, we will start offering this testing panel in our state system, which means that this will be the largest breast cancer panel that is available to state patients, with about 14 genes on it that all are associated with an increased risk for developing breast cancer. In private, the options are a lot wider, but also financially onerous on the person, as not all medical aids cover them. So there is testing for founder mutations. There's also full sequencing that you can do for BRCA1 and BRCA2. You can do single gene testing, and there's also breast cancer panels. So there's panels that you can do locally through certain labs where you can assess multiple genes at once, or there's testing that is available overseas. And so testing locally is often covered by medical aid through your medical savings. For some medical aids, others, they don't cover it, and overseas testing is not considered at all for remuneration by medical aids on our side. Okay, so last little bit, and I guess the gist, I've mentioned a bit about my role and what genetic counselors do, but this is why this is also important and why genetic counselling is important. Genetic counselors, such as myself, are part of your healthcare team. So genetic counselors are healthcare professionals who have specialized education at the master's degree level in both genetics and counseling. So we provide risk assessment, education, and support to individuals and families that are affected by or at risk of genetic disorders. And genetic counselors interpret genetic testing results. They also provide psychological support and serve as patient advocates. So why is it important to get a full family history? Well, we've discussed this and we've touched on it. But as genetic counselors, allows us to accurately assess the risk of cancer in a family, can also provide an indication of the inheritance pattern, so how this cancer is being passed down in the family, but also may guide genetic testing. And as a counselor, we want to aid in the prevention and management of genetic disorders. So there are options. If people know their genetic information, there's ways that they can prevent and manage their risks going forward. We can also help facilitate the appropriate discussion of medical information to assist that people that are making these very challenging decisions are doing so because they are well informed and feel empowered to make these decisions for themselves, but also helping to assist with the patient's psychological well-being. So we are there to help support, to advocate, but also we understand, having seen so many people go through this process, we understand how challenging this decision is, but also how different it is between people. Just to comment, when I was in training, my master's research that I did focused on the psychological impact of undergoing predictive testing for hereditary breast and ovarian cancer syndrome. And so this is something that we all take very seriously, and it is something that's very important to us, which is why we advocate for genetic counselling for people going through this process. And then lastly, contacts and referrals. So I said that we are based in Braamfontein at the National Health Laboratory Service. I've included our details here. This is our landline and call phone. You can also contact us on email if you have any questions. And we also are on Instagram. And if anybody has any questions, they can get hold of me directly. My email was on the front slide, but we can share it. I can get Jenna to share it as well. And then if anyone wants an information leaflet, so we normally have these at the hospitals, but I know that not everyone is necessarily able to access them. I can also send them through to anyone if anyone needs. So I hope that was helpful and informative.
Great. That was really good. Thank you so much. That was great. We've got some questions here. So Nicole asks, can you please discuss which primary cancers are genetically related to breast cancer? Only female reproductive organs, all female reproductive organs, other organs?
Okay, so again, it depends on what genes are involved. So for BRCA, we know that it's going to be ovarian cancer that we look out for. But for other genes, like the moderate risk genes, just off the top of my head, PALB2, for example, is one of the genes where we know that there is an increased risk for liver cancer and pancreatic cancer. And so when we look at a family history of cancer, we try and put all of those things together and try and see what direction we want to go in for testing, which is why these gene panels are so helpful, because sometimes it's not entirely specific. A lot of them do have ovarian cancer, not just BRCA, but also, let's say, for example, Lynch syndrome, where there's also risk for colon cancer, for example. So we want to be able to include a lot of genes where a lot of the cancers overlap at some point. But it does depend. Another gene, TP53, you get a lot of other types of skin cancers and neurological cancers that you don't necessarily get in the others.
Right, thank you. That's clear.
So that helps. Hey, Nicole, answers your question.
Yep, thank you.
Great, cool. Lisa asks, is it possible to do the sequencing without knowing the exact mutation my mother had?
Yes. So if someone comes to us and they know that someone in the family hasn't been has been tested, but they don't know what the mutation is. Again, the testing is, so if it's predictive testing, we'll test for a specific mutation. But if you don't know what the specific mutation is in the family, we do full comprehensive sequencing. So basically reading your genes like a storybook from the beginning right to the end. And if we do find a mutation, then we assume that that was the mutation that was elsewhere in the family. So yes, depends on who's been tested, if anyone's been tested, and then we make that decision from there.
Great, thanks. Okay, Tasnim, you had a comment. Are you there? Do you want to let us know?
Thanks, Jenna. Thank you. I want to just say thank you. I think the session has been exceptionally informative. I've been on the journey for 17 years, as I indicated earlier. I've never ever been genetically tested ever, but I can tell you that from my family history, my mother's sister and my father's sister both had breast cancer. So clearly the gene did come from there, but I personally have never been BRCA tested, BRCA1 or BRCA2. However, having said that, my family going forward is what I'm interested in. And my son at 25 was diagnosed with testicular cancer 5 years ago. So clearly, this family sits with a lot of genetic irregularities. So going forward for me, and this is where I find the discussion very interesting, because obviously I have 2 more sons, I've got a granddaughter, my brother's got kids, and yeah, so going forward, I think it's definitely something that I would definitely look for more information to see how we can actually do testing for family that has not been affected yet and what the risks are going forward. So very informative. Thank you very much.
Thank you, Tasneem. And yes, I see a lot of people in clinic that have a story like yours where they've had cancer for many years, but now all of a sudden there is a concern and wanting to understand the risk for other people in the family. And then typically when we do offer testing, we want to offer testing on someone that has had a cancer. So we don't normally test people that are unaffected. And that's purely because if you haven't developed a cancer and you're unaffected, you could be one of those 2 out of 4 options that haven't inherited a genetic fault. And so you wouldn't be at a higher risk for developing cancer. At least if we're testing someone that has had a cancer, we know that the chances of us finding a mutation in that person are a lot higher. And there's also, we have risk assessment tools that we can use, and there's certain rules and things that we can put together to try and figure out what the chances are of finding a BRCA mutation versus something else. But very often we lean more towards doing panels because of these kinds of things.
Great, thanks. I see, Shana, you have your hand up. Do you have a question or something? Shana, are you there?
Okay. Good morning. Can you hear me now?
Yeah, thanks.
Thank you for a very informative session. I'm quite new into this. Only started my process in 2018, so it's fairly new. I actually looked at my report now while we were talking. My question is twofold. How reliable are these tests? Because my mum was adopted, so we don't have any background around her. Would that have impacted my test results? Because the result is negative for both 1 and 2. But then there's something clearly on the day I went alone. It was up and down. I'm actually looking at this thing for the first time, and I can't remember what was said during the follow-up appointment. But I see on the report there's something to do with K3226X variant. If you can explain that to me. But like I said, is this reliable, seeing that I don't know half of my genetics?
So, Shana, thank you for the question. And I think it's very important in that it sounds like, and if you're comfortable, you're more than welcome to send me that report and I'll have a look at it and I can chat to you about what is on there, because I'm not 100% sure what type of testing. It sounds to me like they've done tumor testing and they haven't done germline testing, is what it sounds like. So,
Monica, just to come in, it was at the state facility. So maybe, yes, that's good.
Yeah. So I'm happy to have a look at it. But we know that even though your mother was adopted, we know that her genetic information inside all of her cells is hers, and she would have passed half of that on to you. And so when we test a person's genetic information, we're testing the information inside the cells of their body. So we take blood typically, and then those cells that are in the blood, we break them down and we capture the person's genetic information, their DNA. That DNA is then read like a storybook in the genes that we are specifically looking at to try and see if there are any faults or changes in that DNA. And so where your mother being adopted comes into play is that we know that if we know that there's a mutation in you, we know that there's a chance you might have inherited it from her. But regarding her family, it would be difficult then. Her results wouldn't apply to the rest of her family, the family that adopted her. They would apply to the family that she was adopted from.
Yes, yes.
Yeah.
Okay, great. Thanks for that. Thank you, Shauna, for sharing.
You missed
Okay,
Nicole's question right in the beginning.
did I?
Yes.
Oh, shucks, thanks. Okay, Nicole, Fuller, in your experience, what is the prevalence in the CDH1 gene, and is it often tested for? Will it be part of the panel?
So I can have a look for you in a second and just comment on whether the CDH1 gene will be part of our panel. But in the meantime, I know that CDH1 is not common. It's not a common mutation, not commonly seen in our breast cancer patients. But again, I'm saying that very reservedly in that because we haven't been able to offer testing on a large scale before, where we've only ever offered BRCA1 and BRCA2, it makes sense then that we wouldn't see it commonly. I'm just looking here. Sorry, the panel
Sorry,
list.
if I can just say, because it's more prevalent in lobular breast cancer with the ECAD, the production of the ECAD heron.
So it is on our panel. CDH1 is on the panel that we are launching in our department later this year. And so, yeah, that will be available to state patients as well. It is on the testing that we offer to our private patients, the patients who pay for overseas testing. It is also there. But yes, again, the different mutations are or different genes are associated sometimes with different types of cancer. So yes, the lobular breast cancers, we see the CDH1 mutations.
Okay, great, thanks. Louise asks, do any of these breast cancer genes affect other diseases or increase the development of other diseases?
Good question, Louise. Yes, sometimes. So conditions like mutations in like TP53, for example, is one where you have a lot of associated neurological problems as well as the cancer risk, but not not BRCA, for example. BRCA, we don't typically see any other conditions that develop. There's other genes like SDHB, for example. I recently saw someone who had a mutation in the SDHB gene, and why we tested this person was because they had a tumor on their kidney. And we know that SDHB, depending where in the gene, so if you think of the gene as being the storybook that I keep on saying, if the mutation occurs on page 3, then you are at risk for like one set of issues. If the mutation occurs on page 103, you're at risk for a different set of issues. And so the mutation in this person increased their risk for kidney cancer, gastrointestinal cancers, but also something called paragangliomas, which also impact the way the body deals with your flight or fight reaction. And so this person had high blood pressure, migraines, a lot of other things that were not necessarily cancers, but also impacted the way their body is functioning and working. And so, yes, in some cases, so this SDHB gene is on the breast cancer panel, but not the mutation that this person had. So it can be quite complicated, but the answer is yes in some cases.
Great, cool. Lou, does that answer your question? Yes, cool. Okay, Heidi, does the full panel of DNA analysis, biotech testing cover what has been discussed this morning?
I'm assuming I'll have to go back and look at the panel because it does get updated every now and then. I know labs like Ampath, for example, have got like a 9-gene panel where I think all the panels that are available in South Africa have the BRCA genes on them. Sometimes they cover a few additional genes depending on how the testing works. One thing to caution everybody on is when you're making a decision about panels, this is why having someone like a genetic counselor in your corner is important, because sometimes the testing looks for, depending on the method that they're using to look for genetic faults. I keep on talking about it as reading it like a storybook from beginning to end, where they're doing sequence analysis. So they're reading one step at a time to see if there's any faults. But what the labs also do is they do deletion and duplication analysis. So what that means is they're looking for long stretches in this book where whole pages have been copied or whole pages have been deleted. And sometimes when you do sequencing and you're reading it like a storybook, you don't realize that there might be whole sections that are repeated. And so depending on what type of testing method they're using, some labs do sequencing and deletion and duplication analysis, whereas others only do sequencing. And so you can rule out spelling mistakes if you're reading it one at a time, but you might not be able to rule out whole additional copies or whole missing copies in that gene. And so I can have a look at it, Heidi, if you want, and we can see what's been offered in that panel and what the shortcomings are, if anything.
Great, cool, thank you. Okay, anyone else with questions? Oh, this has been so informative. Anyone else with questions? I'm gonna do gallery view. Are we all good and happy and had all of our questions answered? Oh, one more, cool. Okay, Heidi's saying, yeah, that would be amazing. Lou? Yeah?
I'm coming.
Yeah.
So with all the genetic testing that's been done, is there possibilities of treatment changing for those patients? A lot of the newer chemo drugs target certain things like TDK4s, etc. Do you think it's going to make a big difference in the long term?
I think in the long term, yes. So the field of genetics that has sort of arisen out of this is what we call pharmacogenetics, where there's drug manufacturers and people understanding how drugs work and trying to link that back to a person's genetic information. So seeing if you have a specific genetic combination, or if your genes have this specific sequence, then you will respond in this kind of way to a drug versus someone who doesn't have the same sequence and they'll respond in that kind of way. And so I do think that it's a very expensive and difficult task that people are trying to undertake. But I think that if you can analyze populations and you can see a lot of people that have very similar genetic makeups within certain genes or certain bits of DNA that are associated with cancer risk, then you can start to develop drugs that are more tailored according to that person's genetic information. The one big downside is that that means that for you to access that kind of treatment, you would have needed to have genetic testing beforehand. And I just don't think that like base system is in place just yet. But I do think that it's heading that way. And there is a lot of benefit in having a treatment that's more tailored towards you. So I do think it will be helpful in the long run, but I think it's going to take a long time.
Okay.
Jen, can I quickly ask something to Monica? One of a lady that I know who has metastatic breast cancer, she has been treated at Donald Gordon, and the chemo regime she was on now had stopped working, and she had more mets to her liver. And Prof. Moodley at Donald Gordon is doing genetic profiling on her liver biopsy to see now what the next line of treatment is. So, I mean, it seems like that they are already doing that a lot, where they're profiling, you know, to tailor the treatment very specifically.
Yeah, and if you think about it, I'm sure a lot of you will be familiar with the system of looking for estrogen receptors and HER2 receptors and all of those. So that was a very sort of the first type of tailoring of treatment, in that they were looking for certain proteins in these cancers, and if you've got an estrogen receptor positive, then you'll respond to this type of treatment. And so that concept has been around, and I get that this is just advancing a little bit more in being able to analyze tumor tissue more intensely with more genetic probes, or trying to understand it a bit better. Sorry, everything is ringing now. But the tricky part about it is that, again, it's only available to a select few people, and it's only testing the tumor. I think what Louise is also was asking in the previous question is, when we start to understand more about the person's germline genetic mutation, so what they were born with, and how can we tailor their treatment already from the outset. Once a cancer develops, how do we prevent it, or how do we manage it very, very early on?
Just to comment, there's a lot of new, very expensive chemo drugs that are available that are not generally covered by the medical aids. And a lot of the genetic testing they're doing is to say, this patient has ABC1 mutation, and this new drug, whatever it is, green highlighter, will work specifically on that. So sometimes they do it to get the medical aids to pay for the very expensive treatment. It doesn't mean that everybody's going to get that treatment. In government, you won't have access to the very expensive drugs. So sometimes there's method to the madness doing these genetic tests to motivate for the 2 million rand drug that can make a difference for you, if that makes sense.
Yeah, and if you think about it again, that's explains why it's so expensive, because the amount of money that goes into developing a drug sometimes, and we've heard stories of drugs for a lot of different conditions that have been developed for 1 or 2 people. There's only 1 or 2 people in the world where that drug actually works on, and it's because that's been funded by parents or families that have really thrown a heckload of money at developing these drugs. But again, if you think about it, then yes, we can manage the treatment, but sometimes if you test someone before they've even developed a cancer, you can manage their risk from the outset without having to spend millions on a treatment going forward. So there's a lot of different approaches, and I guess the end goal is that we want to try and prevent breast cancer and manage it, make it more survivable. Yes, Louise.
Do you think there will be, I don't know if it's available at the moment, any way to change those mutations once you've got them?
Yeah, there's a lot of technology that they've been fiddling around with. So a lot of people would have heard about CRISPR, which is a gene editing tool that people were using. It's a biological tool. So basically what you do is they insert this CRISPR molecule into your cells, and it's supposed to be able to go into your cells, find its target, cut it out, and replace it with an unfaulty copy of that gene. The challenge is that CRISPR is still not good enough to not make mistakes. So even though it's targeted, it's still making errors, it's cutting in the wrong place. And then you wonder, the other devil's advocate side of it is that if you're cutting out this piece of DNA, what other function does it have besides what we know? So we know that it's implicated in breast cancer, but is it serving another important function in the body that we just don't understand yet? So there is gene editing happening, and I think that we are on the forefront of it. I think it's going to explode at some point, and there's going to be a ton of it. But there's, again, a lot of the ethical sides of it need to be approached with caution as well.
Absolutely. Cool. Interesting. Okay, anyone else? Any questions? Last questions? No, are we all good? Okay. Well, thank you so much, Monica, for the session. I feel like
Thank you for having
my
me.
brain is just so happy this morning with all this new knowledge. It's been a great way to start a Saturday. So thank you so much. This was really, really informative. Ladies, if there's anyone that wants to get in contact with Monica or anyone at her team, please just let me know and I'll send out her details, her email address. Otherwise, everybody have a really, really great Saturday. And again, Monica, thank you so much. This was really great.
Such a pleasure. Thank you so much for having me.
Wonderful. Thanks, ladies. Have a happy weekend.
Thank you. Bye,
Thank
everybody.
you.
Bye, everybody.